Thank you very much. I'd like to start by thanking the organizers for this nice invitation. It's really an honor to be invited to such a good seminar this afternoon. We'll start the laser pointer. So as Melanie explained, I'm a group leader at Institut Pasteur at Lille. I'm working most closely with Bart Stales, and my research tries to understand the links between metabolism and immunity and the pathophysiology of NAFLD. And what I'll talk about today is how insulin resistance plays a strong role in all that. I think it fits pretty well with the topic of today's seminar. And and then I'll finish with some of our own work about looking at new molecular links between NAFLD and dyslipidemia. So I have no potential conflicts of interest. All this work has been financed by public funding mechanisms. So to begin, I'd like to talk a little bit about non-alcoholic fatty liver disease. So I'm aware now that, as most of you are as well, that there's a push to rename non-alcoholic fatty liver disease as metabolic associated, or MAFLD, metabolic associated fatty liver disease. For the talk, I'll stick with this older terminology because it's really it has clear clinical criteria that I'll talk about here right now. So NAFLD is really describing a spectrum of diseases which is best characterized by steatosis. So the first step is called fatty liver, non-alcoholic fatty liver, NAFL. When we have inflammation and can have fibrosis, we're starting to talk about NASH, so non-alcoholic steatohepatitis. And then in later stages, we can get to kind of end-stage liver disease, cirrhosis, and can have have these outcomes such as type 2 diabetes, cardiovascular disease, and hepatocellular carcinoma, the latter two being the two most common causes of death in these patients. So as I mentioned, these are really defined clinically by histological analysis at the early stages, steatosis, isolated steatosis with varying amounts of inflammation or ballooning. And once we get to the pathological stage of NASH, we really have the presence of steatosis, ballooning, and inflammation at the same time. So what we know about this is that it's a very heterogeneous disease process. This was taken from a review that we wrote about 4 years ago now. But I think the concept that's trying to be conveyed is that we have patients that can start with only steatosis and can proceed very quickly to advanced NASH and fibrosis with significant fibrosis, whereas certain others may maintain kind of a mild NASH or steatosis-like phenotype for decades even and need no further intervention. And it's really not well understood what are the signals that are driving this, these different tracks of the disease among different individuals. But one of them that we're going to talk about today is diabetes and metabolic dysfunction. So NASH and diabetes, or NAFLD and diabetes, have a special relationship, as I like to put it, because if we think about in the general population, estimates are around 20 to 30% NAFLD, depending on where the study population is, and less than 10% of the population may be affected by NASH. Whereas if we start to talk about the type 2 diabetic population, and especially with obesity, now we can get to 80, 90% NAFLD, excuse me, depending upon how they diagnose it, and up to 20 or 30% of NASH. So this really demonstrates a really key role for metabolic dysfunction and insulin resistance in the development and driving the progression of non-alcoholic fatty liver disease. So we start, if we talk about NAFLD, as I mentioned, one of the first steps is the steatosis. And I'd like to give kind of a brief overview of the major ins and outs of hepatic lipid metabolism. And what we know now about how the liver gets fat. So the major inputs here are coming from free fatty acid uptake, mostly from the adipose tissue in the diet, chylomicron or LDL clearance, lipogenesis, which is now recognized to play a major role in the development of hepatic steatosis. And then in terms of outputs, we know that VLDL secretion, hepatic VLDL triglyceride secretion is one of the major outputs, and to a lesser extent, ketone body production as a result of fatty acid oxidation. So in the context of NAFLD, as I mentioned, everything that you can see here in yellow is strongly upregulated, and I'll talk about some of the connections as to why in a little bit. But we know from some elegant studies from Ken Donnelly over 20 years, almost 20 years ago now, that the adipose tissue NEFA are a major component of the fatty acids that end up composing triglycerides in the context of NAFLD. So I just want to talk a little bit about why that is. So as most of you are aware, one of the adipose tissue is the major fat storage depot in the body, and during fasting we have a lipolytic signal that drives the release of free fatty acids, which are recovered by the liver and esterified into triglycerides by DGAT. In the postprandial state, insulin is normally supposed to block this free fatty acid release. So as our first speaker tonight, Andre, spoke about earlier. But what happens is that in insulin resistant state, now we no longer have this suppression of free fatty acid release, and we can start to get abundant free fatty acids coming back to the liver, which we'll just take them and store them as efficiently as we can. And so you can see that we know that this is important in NAFLD, not just from these previous studies, but we have this nice data showing the effect of adipose tissue insulin resistance in NAFLD. Here we're looking at the suppression of free fatty acid released by insulin during a clamp study. And we show that as we increase the plasma insulin concentration, free fatty acid generally goes down, but in the NAFLD group, these patients maintain higher levels of free fatty acids, and this will translate of course, into higher intrahepatic triglyceride content. We also know from some studies from Rudy Zechner's group in mice that if we block adipose ATGL using a pharmacological inhibitor, that we can reduce the hepatic steatosis component here in mice, wild-type mice fed a high-fat diet. So clearly, this adipose tissue free fatty acid can play an important role in the development of steatosis in the context of NAFLD. So the next thing that I wanted to talk about is this other major component that's getting more and more interest now as several groups have been working on tracer studies and shown that lipogenesis is actually a major component of a major disease feature and also a major driver of lipid accumulation in the liver. So as most of you are aware, insulin in the liver has two major roles, one of them stimulating lipogenesis and the other blocking gluconeogenesis. And so, of course, in insulin resistant states, we have this kind of paradox where the lipogenic pathway remains sensitive to insulin. And so hyperinsulinemia are driving lipogenesis excessively, whereas gluconeogenesis is no longer sensitive to insulin signaling. And we have this setting where we have hyperglycemia in the context of hypertriglyceridemia as a result. And just to give an example of the magnitude of the contribution of de novo lipogenesis to this to the phenotype in NAFLD, here's one of the more recent studies from from Sam Klein's group, looking at the contribution of lipogenesis, de novo lipogenesis to the fat content of VLDL. And we see that in lean patients, we have around 10%, but in obese patients, we can get up to 40 or even 50% of the total triglyceride content of liver-derived lipoproteins as being a result of lipogenesis. So it's really demonstrating this huge increase in activity of lipogenesis that's associated with NAFLD. So now I've spoken a little bit about these hepatic lipid metabolism and how insulin resistance can lead to the increases in triglyceride. And one of the last things that I want to touch on before I get to some of our own data is this link between steatosis and especially atherogenic dyslipidemia. So I'm going to focus today on the link with serum triglycerides and hepatic triglycerides that have been studied very nicely recently. So this is a kind of an older paper from the group in Gothenburg, where they looked at the hepatic VLDL triglyceride output as a function of plasma glucose levels. And this was really to show that the increase in hepatic triglyceride output is very closely associated with an increase in plasma glucose level, and it's actually a key feature of diabetic associated dyslipidemia. And at the same time, we have another study from Sam Klein's group looking at the hepatic triglyceride secretion rate again on the y-axis, but this time as a function of intrahepatic triglyceride content. So here we have on one side hyperglycemia that we can consider an index of hepatic insulin resistance. And then on the other side, intrahepatic triglyceride content. And in both cases, we see a very strong positive correlation with the concentration or the rate of hepatic triglyceride secretion. So these two factors together can drive one of the major features of atherogenic dyslipidemia. So just to wrap up in this first part, I'd like to just remind that non-alcoholic fatty liver disease is really more and more considered the hepatic manifestation of the metabolic syndrome. It's related to insulin resistance in multiple tissues that leads to hepatic steatosis. And related to both of these factors together are combining to drive altered hepatic lipoprotein metabolism. And so I think one of the important things that will come forward is trying to understand what are really the molecular links that are driving these connections. So now I'd like to kind of switch gears and talk more about a little bit about a recent study that we've done. We've had the chance to work with two great clinical collaborators with some complementary NAFLD cohorts. One being the Resolve cohort that was started as part of an FP7 project with Sven Franke in Antwerp. And the other was one of our local collaborators here at the EBOS cohort with François Patou. And what we have is hepatic transcriptomic data paired with liver biopsies, where we were able to perform transcriptomics on these patients. And the real power of these cohorts is the work of our clinical collaborators, where they've done a really great job phenotyping these patients. And so we started with just a simple question of what are the liver genes that are expressed that change with the level of steatosis and that may also be correlated with the plasma triglyceride levels. So if we started first with the EBOS cohort, and we found this interesting lipoprotein called apolipoprotein F. So here on the left, I'm showing you the correlation of the plasma triglyceride levels with the hepatic ApoF expression. This may not look that impressive, but it's actually the second most strongly inversely correlated gene in the transcriptome with of the liver transcriptome with plasma triglyceride levels. And it also strongly decreases with increasing steatosis grade in these patients as well. So between these two things, we see that ApoF is decrease in ApoF is strongly associated with plasma triglycerides and steatosis. And this is, again, these are among the strongest associations in the entire transcriptome. With our collaborators from Antwerp, we were able to confirm this correlation. The plasma triglyceride levels are not quite as exaggerated as in the EBOS cohort, but we still see the same trend for an inverse correlation with hepatic ApoF expression and plasma triglyceride levels. And then on the right, again, the correlation with hepatic ApoF expression and steatosis grade as measured by histology. And again, it was really surprising because ApoF is one of the strongest inverse correlated genes with hepatic steatosis, but it's something that we haven't really heard a lot about. So I'll take you through a little bit about what we know about ApoF first. So ApoF, it's a highly specific to the liver. It's over 10,000 or a million-fold higher expressed in the liver than any other tissue that we've studied. It was originally identified by Sven-Olof Olofsson and as a component of HDL and to a lesser extent LDL. So I'll give a brief overview now about lipoprotein metabolism as it pertains to what we're going to talk about today. So I'm going to focus mostly on VLDL and LDL metabolism. I won't talk much about HDL. In terms of functional correlation, there have been some broad studies about just correlating the ApoF, plasma ApoF levels that found that plasma ApoF is strongly positively correlated with HDL cholesterol, which makes sense as a component of HDL. It's also inversely correlated with plasma triglycerides. There have been 2 studies that have shown that. In terms of functional studies, it was originally identified as an inhibitor of the CETP enzyme, but later studies haven't really proved that to be very robust. The only thing that have really been found in mouse studies this far it was a study from Dan Rader's group that found that ApoF overexpression reduced plasma total cholesterol by enhancing the reverse cholesterol transport. But any links to VLDL and LDL triglyceride-rich lipoprotein metabolism haven't been described. So we wanted to investigate this link between, again, steatosis and plasma triglycerides. And so we started by overexpressing the human ApoF isoform in mice. And as a surprise, we looked and we found that plasma triglycerides were decreased in these mice with no real effect on the plasma cholesterol. I'm not showing that data. And as you might expect, when you look at the lipoprotein profile of these animals, we can see decreased triglycerides. So here in the x-axis, we have a measure of the plasma triglyceride content. And then on the y-axis, we've separated these using a size exclusion chromatography, and we can separate the different lipoprotein classes. And we see that in each class of lipoprotein, we have significantly decreased amount of triglyceride in the ApoF overexpressing mice. So if we look at the plasma triglyceride levels. And of course, this is going to be a balance between the hepatic output, which is managed by secretion, and the peripheral clearance, either by lipoprotein lipase, which can be negatively regulated by ApoC3, or the clearance of the clearance of the remnant-like particles, or conversion to LDL. So as a first step, we wanted to try to dissect whether ApoF would be affecting more the clearance or the secretion pathway. So to do this, we started with a classical assay where we block lipoprotein lipase through the injection of a detergent. And then we measure the production of triglycerides over time. Here's the results of these experiments. So at T0, we inject our inhibitor, and we can see that the accumulation of triglycerides is more or less constant after 60 minutes. And so what we see is that over time, the ApoF overexpressing mice actually accumulates slightly more triglyceride in the plasma, meaning, and because we've blocked the clearance, this means that the hepatic output of triglycerides is increased in these mice. And we can calculate that here. I've calculated that here on the right for you, and it's as a proportion of the slope. It's related to the slope of this line here. So if you're keeping track, now we have plasma triglyceride levels that are decreased, but secretion levels that are increased. So if we keep track, somewhere along the line, there has to be an increased clearance, either by lipoprotein lipase or this on the remnant side. And so we, thanks to a collaboration with Patrick Renson and Sander Kooijman in Leiden, we were able to directly test these clearance assays using the radiolabeled triglyceride-like particles, triglyceride-rich lipoprotein-like particles. So how this works is we inject the particles at a time 0, and then very fast, because mice have a very fast triglyceride turnover metabolism, we can measure the plasma, and we measure the decrease of both the triglyceride content and the cholesterol content, the radiolabeled content in the plasma. And then at the end, we sacrifice the mice, and we can measure the incorporation into the different tissues. So when we're looking at because the triglyceride is labeled on the fatty acid with tritium, we can distinguish whether the tritium decrease and whether it's going into the different organs faster as a result of ApoF overexpression. And this would be indicative of a change in lipase activity. So here are the results from the study. We found, if on the left, we have the curve of the change in plasma tritium. So this, again, related to the fatty acids and the triglyceride, is decreasing over time. And these curves are essentially superposed with whether we're overexpressing GFP or human ApoF. And if we look at the incorporation into the different tissues, we don't see any significant differences in the uptake, especially in the intrascapular or subscapular BAT, which would be the major targets of free fatty acid here in this assay, and no change in the liver. We also measured the lipoprotein lipase activity ex vivo using from plasma, post-heparin plasma in mice overexpressing this. And in keeping with our results from the in vivo assay, we found no differences in either lipoprotein lipase activity or the hepatic lipase specific activity. So then if we go back to this assay, we can actually measure at the same time the clearance of the remnant-like particles as a result of the incorporation of 14C cholesterol in these lipoprotein particles that we've injected. So now we can see that if we do the same assay and then measure the cholesterol changes, we can get an idea about the speed of remnant clearance here. And sorry. And here are the results. You can see that in the ApoF overexpressing line in orange, we have a modestly faster decrease at all time points along this curve, indicating that the clearance of the total clearance of these particles is faster in the ApoF overexpressing mice. And this is also reflected by the increase in 14C cholesterol that we can see accumulated in the liver. So again, pointing to the target of the liver as the major clearance organ for the cholesterol remnants. Sorry. So now we can say that ApoF, this highly liver-specific transcript, which seems to be decreased in NAFLD, but at the same time favoring the clearance of the triglyceride-poor remnants. And so we wanted to go a little bit further to try to understand what could be the mechanism of this, especially on the clearance side of this equation. So we used a transcriptomic approach where we took the livers of our mice overexpressing ApoF or GFP, and we measured using Affymetrix transcriptomics. We identified a number of differentially expressed genes. If we look at the genes that are increased in the ApoF overexpressing model, and we find among the top 2 hits in this pathway are genes related to cholesterol synthesis and cholesterol metabolism. So I realized that cholesterol synthesis and cholesterol metabolism are somewhat counterintuitive if we think about the synthesis side of this pathway, but this is also highly redundant with the clearance side of the pathway as well. So here I've summarized our results from of these genes. We have the SRABP2 pathway, which is the major regulator of cholesterol synthesis. It's also the HMG-CoA reductase, which is the target of statins. And actually, statins work by blocking HMG-CoA reductase, which upregulates all the rest of this pathway, leading to an increase in LDL receptor activity and favoring clearance. And so what we think we have is something a similar mechanism whereby we activate this pathway. We're not really sure how we're eliminating, we don't have all the mass balance worked out quite yet, but it seems to work through this pathway. And we can also see changes in some of the other associated remnant receptors here. We have increase in LRP1 transcript levels, increase in syndican-1, no changes in LRP5 or LRP10, and no differences in other major regulators of the LPL activity, which again is consistent with the idea that we don't have an effect on lipase activity, but more likely something in clearance. So we also have some changes we also have increases in IDOL and PCSK9, which are not really consistent with increased activity of LDL, but we think that may be related to a net effect of the increase in LDL, but this needs to be worked out further. So to test whether this could be a cell autonomous effect, we switched to an in vitro model where we looked at immortalized human hepatocytes, which are a non-cancerous human hepatocyte line. And we wanted to look at a classical model of VLDL or LDL, in this case, uptake. And so we can treat these cells with siRNAs and reduce ApoF. And we found that if we measure the uptake of a fluorescently labeled VLDL particle, now when we reduce ApoF expression, we also have a reduction in LDL uptake. So this again consistent that ApoF activity may somehow affect LDL uptake activity. And this is also related, we think this may be related to the something related to the efficiency of the recycling of LDL at the membrane. Again, if we knock down ApoF in these cells, here we're looking at using a FACS assay to measure the extracellular surface membrane LDL protein. We can see that here in red, we have the control, and in blue, we have a shift. This is again a log scale, so we have a shift of about 50% decrease in membrane-associated LDL protein, LDL receptor protein, compared to nearly an 80% if we use the SI against the LDL receptor itself. So we're talking about a pretty significant shift in the LDL receptor activity. So just to finish quickly here, and then we can take your question. I'd be happy to take your questions. I think we've identified an interesting link between NAFLD and dyslipidemia, and think that ApoF is favoring hepatic triglyceride exports for peripheral storage because we increase secretion. Of course, then all these triglycerides can be stored in the peripheral tissues thanks to the activity of lipases. And then we also have an increase in the clearance to try to get rid of these particles from the circulation. So with that, I'll conclude. I'd like to thank all these people, of course, who have participated in the study, especially Audrey Desprins, who is my PhD student who's been doing most of the work on this study, our clinical collaborators in Antwerp and in Lille, led by Sven Franke and François Patou, a group of Dan Rader, and we've had lots of help from the people in Groningen to work with mouse models and in light, and also for the lipoprotein kinetic measurements. Thank you very much.